I want you to understand the big picture, and now we're going to get into the details where it looks a little scary here, but so I'm going to just tell you what you need to know. There are really about 3 things you need to know, maybe 4 things. I sometimes add a 4th thing, but the 3 things you want to know is that these green structures are stationary complexes, okay, within the inner mitochondrial membrane, shown as the red structure. These complexes are made up of many components, but primarily protein components and enzyme, at least one enzyme. So these are fixed, in a fixed position in the membrane. Each of them, so the first thing you want to know is that these guys are made up of proteins, usually some prosthetic groups associated with the proteins, and they are stationary. So that's the first thing you want to know. The second important thing you want to know is that you want to understand the flow of electrons. First, you want to know, in that part of that, you want to know where these electrons are coming from. The electrons, think of this blue part underneath as the matrix. Okay, you can write matrix if you want there. You can also write in that matrix NADH and FADH2. So the second important thing is you want to know the flow of electrons through the electron transport chain. So I'll go through it sort of kind of briefly here, because we're going to cover it in more detail in the upcoming slides. But the first thing I want you to see is if you have NADH here in the matrix, it will have direct access to this first complex. Here it is, NADH. And don't take this figure literally, meaning, I mean, this complex could be here, but it's part of the membrane, okay? So that means the NADH here has direct access to the complex 1. The function of complex 1 is to oxidize NADH. So here's NADH. When you oxidize it, meaning remove the electrons from it, you get NAD+. Where are these electrons going? They are gained by this molecule FMN, a type of prosthetic group. A flavine mononucleotide is a prosthetic group that will gain the electrons, and that's why we call it FMNH2. Now this prosthetic group is going to eventually donate its electron to another important electron acceptor called coenzyme Q. And then coenzyme Q has these electrons, so that's why you have the H's. Coenzyme QH2. While I'm here, I just want to say this is a type of an electron acceptor donor that is mobile. So that's really the second important thing, or maybe the third important thing I want you to know, which is that it can, these 4, the green are stationary. This one is mobile. It can move around the membrane. And because of that, it's able to carry the electrons from one complex to the next. So the function of coenzyme QH2 is to carry the electrons from complex 1, let's say, to complex 3. Complex 3 now is going to oxidize, extract the electrons from coenzyme QH, and itself will get reduced, complex 3, and it will actually donate these electrons to cytochrome C here. Cytochrome C is similar to coenzyme Q in that it is a mobile molecule, a mobile carrier. So it's able to move, carry the electrons from complex 3 to complex 4. So cytochrome C gets oxidized, donates its electrons to complex 4, and at complex 4, these electrons are finally given to oxygen to form water. Okay, so you want to know the flow of the electrons. That's an important thing. The second thing, you want to know the difference between the stationary complexes and the mobile electron carriers. Third thing is going to be, always remember that the final acceptor is the oxygen at the end there near by complex 4. And the fourth thing is really what we're going to get into the detail here, is we want to focus on what's in each complex. Okay? So each complex is going to have at least one enzyme and one or more prosthetic group. And you want to think of these prosthetic groups as important carriers of electrons. They are physically involved in moving the electrons. So each of these complexes will have at least one enzyme, one or more prosthetic groups you want to know. And that's what we will cover here in the next several slides. Okay, so here's the first complex. The enzyme in the first complex is NADH dehydrogenase. So it's pretty self-evident. That means it's going to oxidize NADH and donate the electrons to FMN, then FMNH2, then that does donate them to coenzyme Q. Okay? But let's move to complex 2. Complex 2 has succinate dehydrogenase. You say maybe if you're up to date on what we've covered, you say that sounds familiar. I say you're correct. Okay? So that's the same enzyme in the TCA cycle. If you haven't heard of it before, then you're behind. So that's the same enzyme in the TCA cycle. It's really part of the enzyme is in the membrane, but it is extended into the matrix. So it serves a function in the TCA cycle as well as in the electron transport chain. We'll talk about that in a minute, some more. Complex 3 has cytochrome C reductase, and complex 4 has cytochrome C oxidase. So these are the green structures in the previous slide. They are stationary. They don't move. They're fixed. But between these different complexes, there are two mobile electron carriers, coenzyme Q and cytochrome C. Coenzyme Q works between complex 1 and 3, carries electrons between complex 1 and 3, or 2 and 3. Whereas cytochrome C can carry electrons between complex 3 and 4. So the second important component of all these complexes is what prosthetic group is there. So complex 1 has this FMN, which is really derived from riboflavin. Both of these are derived from riboflavin, so they're vitamin derivatives. FMN and FAD are respectively part of complex 1 and complex 2. So they play a role in accepting and donating electrons to the next recipient. Complex 3 has a heme group, but it has a ferric heme group, Fe plus 3. And complex 4 has a copper and a heme group, okay, as the prosthetic groups. So again, these are important in physically accepting the electrons, but remember that what catalyzes that exchange are usually the enzymes in these complexes. The prosthetic groups on their own can't do it. They need the respective enzymes in each of the complexes. So I'm going to remind you now how we begin with electron transport chain and what we need. Like I said, you have to remember that the function of electron transport chain is to oxidize NADH or FADH2. Both of these, for the most part, again, underscore most part, are produced in the matrix. The electron transport chain is right adjacent to the matrix in the inner mitochondrial membrane. So they both have access directly to the electron transport complexes. And as a reminder, where's the NADH coming from? The NADH is coming from all the enzymes we covered last week. Pyruvate dehydrogenase, right there. Malate dehydrogenase, alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase. In addition, fatty acid beta oxidation produces NADH. So anytime you have a large amount of NADH in the matrix, it will be oxidized at complex 1. Now, there's NADH that is produced from glyceraldehyde 3-phosphate dehydrogenase in glycolysis. We're going to treat that separately because that NADH is produced in the cytoplasm. Okay? So we have to manage it in a different way. So now we go to the other important substrate. So I want you to always think that the substrate for complex 1 is NADH. Only NADH is oxidized at complex 1. The substrate for complex 2 is FADH2. Again, that's produced from succinate dehydrogenase in the TCA cycle and from fatty acid beta oxidation. So you have a lot of FADH2 in the matrix. It will start being oxidized at complex 2. You cannot exchange that. Okay? NADH always at complex 1, FADH2 always at complex 2. So that's what I mean by knowing the flow of electrons. So if you start with NADH, let's say this NADH is produced by pyruvate dehydrogenase. It will get oxidized at complex 1. Complex NADH dehydrogenase in this complex will take these electrons, give them to coenzyme Q. Coenzyme Q is the mobile carrier. It will kind of move around the membrane and goes to complex 3. Note, you skip complex 2, you just go to complex 3. Coenzyme Q is carrying the electrons from NADH to complex 3, where it's oxidized by complex 3, meaning it loses its electrons, and the electrons are eventually at complex 3 gained by cytochrome C. So the enzyme here in complex 3 is called cytochrome C reductase because it's going to reduce cytochrome C, the other mobile carrier, which means that cytochrome C, once reduced, moves the electrons from complex 3 to complex 4, which contains cytochrome C oxidase, removing the electrons from the mobile carrier cytochrome C and eventually donating them to oxygen to form water. So that's the flow if you started with NADH. You may have more FADH2. If you have FADH2 in the matrix, you start at complex 2. Again, the same thing happens. Coenzyme Q is the carrier of the electrons between complex 2 and 3, and the rest of the electron transport steps 2 and 3 are the same. Okay? So you can start either at complex 1 or at complex 2, depending on what you have as a substrate, NADH or FADH2. Okay. All right, you still with me or time for a nap? Okay. Okay, we still have a few things. So here's the big picture. So this white part is the inner mitochondrial membrane. Here's the matrix, and here's the intermembranous space. So here's NADH being oxidized at complex 1. That means the electrons have been donated to coenzyme Q. Coenzyme Q gives it to complex 3. Complex 3 then gives these electrons to cytochrome C. Cytochrome C then moves these electrons to complex 4. So these two are the mobile electron carriers. Coenzyme Q between either 1 and 2 or 1 and 3 or 2 and 3. But note how each time you these electrons, you release protons. And because of the impermeable nature of the inner mitochondrial membrane, these protons are trapped in that space between the inner mitochondrial membrane and the outer membrane, which would be kind of to the top right corner of the slide. So what's going to happen with these protons? These protons are actually going to be the driving force for the second part of electron transport chain, which is oxidative phosphorylation. So the last part occurs or oxidative phosphorylation occurs at complex 5. So think of the last component of, again, this is still the inner mitochondrial membrane. This is complex 5. And as I mentioned to you, it's going to have a dual function. It will act as a proton gate, allowing the protons that have accumulated in the intermembrane space into the matrix. By doing so, it will generate enough energy to allow the synthesis of ATP from ADP and inorganic phosphate. So the dual function of complex 5 is that it's a proton gate, and it is an enzyme that can synthesize ATP synthase, that can synthesize ATP from inorganic phosphate and ADP.
If you like this video, please subscribe. Thank you.